Hi everyone. Today we are discussing about the gel casting and how should I put the condition of gel electrophoresis and which mechanism is working during the gel electrophoresis and how to do the gel lock and which mechanism will be the working and why we get the band intensity and different type of band of the DNA with his mass variation and his length variation using the gel dock. So in this lecture, we are discussing about all these things. So we go through the next slide. So first of all, we talk about the gel casting. So what is the gel casting? Gel casting is such type of the process in which we have the some gel powder and using the some buffer and hitting it and put into the some tray with the comb and easily to segment and put for the some time and easily to get the gel. Okay, so yeah, this is the Agarose gel. We having the Agarose gel for the electrophoresis techniques to separate the DNA molecule according to their size or weight. Actually, which DNA has the larger size, that DNA has the higher molecular weight. Okay. DNA molecules. What do you think about the DNA molecule? DNA molecule have the phosphate backbone. Okay, so it has the highly negatively charged. Okay, so this is the highly negative charge atom always attract to the highly positive charge. So it is the attracted toward the anode side, that is the positive electrode. Okay, and smaller DNA fragment move faster as compared to larger due to the smaller DNA have the smaller size and larger DNA fragment have the higher mass. Okay, so components, many of the components and its function has been discussed here, like as average form the forest network of where DNA molecule is moved from the negative side, that is the cathode to anode. Okay, and running buffer, two types of running buffer we are generally used, like the TAE and the TBE, which is maintaining the electrical conductivity throughout the process. Okay, for the maintaining the pH. Okay, and we have the DNA samples. Actually, that DNA samples molecules to be separated. To DNA sample is a very compulsion things. So otherwise, what you want to analysis during this process and loading die. This is also very important things. Then easily to we track all these things like is like as how much our DNA have been migrated from the initial point. So loading die which will vary also initial part and DNA ladder. Ladder is that type. Ladder is one type of marker that is easily to analysis that our DNA is the actually we get the good correct DNA. Otherwise, we easily to analysis that our DNA is this that length of the DNA, this that mass of the DNA. Okay, and electrical power supply. Okay, this is the very also an essential part of this process, like the electrical power process. Then we easily to create the positive cathode and positive anode and cathode in electrical circuits. So throughout the process, electrophoresis process, electrical power supplied and Ethidium bromide. This is working as a standard with adding during the gel casting. Okay, this is the Ethidium bromide. We are discussing all these things. Why Agarose? Agarose is the mesh-like structure, so DNA is easily passed through that force and that mesh. And the larger DNA experience more resistance and smaller is the less resistant due to the any person is the have any places such of the mesh. So mesh has the fixed size, so bigger size any substances passing through that mesh so has facing some resistance. Okay, so this is the process through of the we are making, yeah, casting the gel, like this is the TAE buffer, and this is the Agarose gel powder, and make the solution and heat, and this is the well to mix and add the standard like as the ETBR and put into the for the casting. Okay, and after the casting and after the gel dock, such type of the result we are getting. So I will discuss about this in some later slide. So then we discuss about why DNA is smooth. Okay, so we know all the person about that DNA is the highly negatively charged. This is the sugar phosphate backbone is here. This is the highly negatively charged. Okay, so this is the highly negative charge, and one one cathode of size is the positive electrode. So this sugar phosphate bond is highly negatively charged is the attract toward the positive anode, positive electrode, and many of the bonds will be showing here. That is the adenine, guanine, thymine, and cytosine. Okay, so here also mention all these things like the DNA backbone contain phosphates group, and is phosphate is the highly negatively charged like is shown in here. So overall DNA is negatively charged. So voltage applies like the cathode DNA to anode. So DNA is migrated toward the positive side. Here is we are loading the DNA samples, and here is the migrated from the negative to positive side. And electrophoresis tank is the buffer solution also put hole over this chamber and moving direction toward this. And here is the electrical power supply. Okay. And here just set something the voltage, current, and time. Okay. So next we will discuss about why we are setting the voltage, current, and time in this process. Okay. So we are apply the voltage during this process near about 80 to 120 voltage. Okay. So voltage is visually experience the electrostatic force, and that electrostatic force to produce to migrated to support the migration of DNA charges. Okay, so this is the support for the migration of DNA, and DNA is moving through the momentum, electrostatic force, and gel friction, so size and velocity, something like it. Okay, so using these things is getting some electric field and then electric field to velocity. Okay, so some common role like as the buffer is also maintaining the pH, carrying the current, and maintain the pH and protect the DNA. This damaging of DNA is preventing, and heating also maintain the heating so easily to carry or temp current. Okay, this is the doing by the running buffer. What's happen chemicals in buffer? Okay? What do you happen chemical in the buffer? So one is our cathode side here, the water plus electron making release the hydrogen and OH, and here another end is the anode side, then water is release the oxygen gas form, and one places the hydrogen. So overall reaction is water is releasing hydrogen and oxygen. This is the electrostatic. So bubble will appear during the electrostatic during this process. Okay. So why we are using the Ethidium bromide, and that is the stanner. Stanner is the our Ethidium bromide. That chemical formula is C21H20N3Br. Is the fluorescent dye. Structure is the planar aromatic molecule. Okay. Slide between the DNA base pair to called intercalate. Actually, that is intercalate between the DNA molecules. So this is intercalating between AT, A T and GC between them. So it easily to intercalate this molecule in between them. So easily to get the some fluorescent. Okay. Intercalating like is that here is the normal DNA at TGC like is first type of the DNA, and intercalating between them subside like this band, this in between ETBR will be founding bomb like the pi stacking and van der Waals interaction, electrostatic interaction like as. So some chemical formula also exist here like the DNA plus ETBR is forming some complex structure like this is happening at the equilibrium reaction. Okay. Like as here the in between the intercalating here this is ETBR. Okay. And at some light intensity it will give the fluorescent under a UV light, give the fluorescent and take the image and give the band. Okay, so yeah, how to glow our DNA? So Ethidium bromide absorb the UV light. So excitation of UV light is absorbed by the Ethidium bromide, and this is going to the excitation state and visible as the orange light at 590 nanomolar. So Ethidium bromide in the free in the solution. So fluorescent is weak. When DNA bond to the DNA, fluorescent intensity increases 20 times. So this such type of the we having. So if we have the free ETBR UV energy loss as a heat weak fluorescent and DNA bonds ETBR then UV restricted rotation, more emitting light and bright fluorescent. Okay. Here is the like as the same mechanism is using this instrument that name is the gel dog. Okay. So entire experiment, first of all, we are preparing the Agarose like as the costing the preparing Agarose powder and adding the wafer and pour into the gel, insert the comb and gel solidify, remove the comb and fill the tank with wafer of the for the electrophoresis process and loading the sample with dye. Okay. Apply the some voltage, current and time, and then DNA is migrated from negative anode, negative cathode to anode, and smaller DNA travel faster and stop electrophoresis. Stain with Ethidium bromide to use the pre-stain in gel. Here we are adding pour gel. Pouring before pouring we are adding the stainer and UV luminous and DNA binds up here. This is the whole experiment. And the summary of the complete mechanism is the DNA is the negatively charged, so electric field applied, so DNA is migrated toward the anode side, and due to why it is the migrated because of the our Agarose is the mesh lies separated structure to DNA easily move throughout. So a small fragment is the moving faster and larger fragment facing the some resistance of that Agarose mesh. So our Ethidium bromide intercalate between the base DNA base pairs and DNA plus Ethidium bromide complex form that irreversible bonding and UV excitation at 300 nanometer and orange fluorescent gives at 590 nanomolar and visible the DNA bonds. Okay. So this is the whole lecture. So throughout we have been discuss about what should I do during the Agarose from the Agarose preparation, otherwise gel casting to DNA analysis. This is the whole process and how the whole process is working and what mechanism is working behind it. So thank you guys attending this lecture. If you have any doubt, so please comment me and ask me freely and I am happy to share the answer of that problem. So thank you guys.